Hi, everybody. My name is Stacy Leonard. I'm the events coordinator here at Connecticut River Conservancy. We're really happy you're all here today with us for LiveStream episode 6. It's been an exciting series of lunchtime presentations we've been offering this spring and summer, where we bring our work and our rivers right to you in your home or wherever you may be hanging out. We hope most of all that everybody is staying healthy and safe during these very uncertain times. I am soon to introduce to you Ayla Skorupa, who is going to be leading our presentation today, but wanted to share a few other details for newcomers. Firstly, I want to announce that we've just scheduled 2 new episodes for August. The August 5th episode is especially suited for kids that we're calling kids ages 2 to 82. It's about fish migration, and it's going to be really fun and dynamic, and it's shorter than our normal series, and it's being led by Sarah Repair, our one of CRC's own staff members. And then on August 19th, we have a special guest, Wick Griswold, who is a naturalist, professor, and an author with the Estuary magazine, who will share insights into the magazine and his writings. So stay tuned for all of that. And at the end of this episode, you'll be getting an email with all the links and easy ways for you to get back to the pages that share all these details. So some Zoom details before we get started. You notice that your mic and your video are turned off, but we do want to encourage you to participate through asking questions. And there is a Q&A box at the bottom of your screen. Take a moment to find that and start asking any questions that you have now or anytime along the way. The presentation will last about 30 minutes and we'll hold all the questions to the end, and Ayla will come on for questions, Q&A, as much as we can get through. If you have any technical troubles, we have Aliki on the line. She is our Zoom guru in the back. And because you may have trouble online, we want to share our phone number to call her, 413-772-2020, extension 207. Okay, I think that's all the details. Just keep in mind the Q&A box and get some lunch and water, and we'll be ready to go. So I'd love to introduce to you Ayla Skorupa, our wonderful presenter for today. Ayla is currently a PhD student at UMass in environmental conservation in the Massachusetts Cooperative Fish and Wildlife Research Unit. Her earlier studies were in marine biology up in Portland, Maine with the Gulf of Maine Research Institute, and then to Fairbanks, Alaska for a master's. She caught the mussel bug, which she'll tell you about, in 2016 and helped to launch CRC's Mussel Restoration Project as one of our early community science programs. In that project, volunteers got to learn all about freshwater mussel and collect data on their habitats. And all of that work, combined with numerous collaborators who she'll also tell you about, led to the creation of a restoration strategy for the Brook floater, which is an endangered freshwater mussel in Massachusetts that Ayla will elucidate us with today. So without further ado, I'd like to turn it over to Ayla, and we'll come back on in another half hour to answer your questions. Thank you, Ayla. Take it away.
Great. Thanks, Stacy. That's a great introduction. So today I'm going to start by telling you a little bit about freshwater mussels in general. And then I will go into this restoration strategy that I've been working on for my PhD with a lot of collaborators. So my advisor is Allison Roy at the USGS Mass Co-op Fish and Wildlife Research Unit at UMass Amherst. And we work really closely with the state, so Mass Wildlife, and specifically Pete Hazelton, who's in the Natural Heritage and Endangered Species Program, and who has since left there to start a job at the University of Georgia. Our federal collaborators are Dave Perkins and Tim Warren at the Cronin Aquatic Resource Center, which is here in Sunderland, Massachusetts. And then Andy Fisk was really instrumental in starting this work with the Connecticut River Conservancy. So if you're going about your daily routine, you probably wouldn't encounter a freshwater mussel. They live on the bottom of streams and rivers and are also found in ponds and lakes. And this is what they look like, kind of just rocks that are protruding from the sediment. They have siphons, so they're actually filter feeders. They have an in-current and an ex-current siphon. And the front part of the mussel is buried in the substrate and is a foot. So they use that foot to anchor during feeding and to move around. There can be a lot of confusion on mussels and freshwater mussels in general. So usually when you hear freshwater mussels, the first thing you think of is the invasive zebra mussel, and that's pictured down here. So this is an introduced species, it's not native, along with the Asian clam. But what I'm referring to are our native freshwater mussels. These are generally larger and they have really unique morphologies and colors, as you can see here. And taxonomically, they're really distinct or distant from the mussels that you go out to dinner and you order and you get on your plate. So those are from the ocean, those are marine mussels. So all the mussels that I'm referring to today are these native freshwater mussels. And as a group or an order, they're called Unionida. And globally, there's about 900 species. They occur on all continents except Antarctica. And they have really fun names like fat pocketbook or rabbit's foot, heel splitter. And on top of that, they have these really cool colors. And here in the Atlantic slope, we don't have as many species as down south. But in North America, it is a biodiversity hotspot, so there's about 300 species here. And what makes freshwater mussels so unique, but also really challenging to study, is their complex life cycle. So for reproduction, there's two mussels, they're separate, male and female, and the female will filter the sperm through, and this will fertilize her eggs. And these eggs will then develop into larvae. And so in the mussel world, this is called glaucidia. And the female mussel now has a job of trying to attract a fish, because these glaucidia are actually parasites and they need a fish in order to develop. So mussels have different strategies for this, but one method is a lure. So they will use their mantle flaps to create a lure that looks like a little minnow. And then, for example, a bass will come and bite this lure, and then the glaucidia will be released and attach all over the fish, or primarily on the fish gills. So here's a picture of these little glaucidia attaching all over the fish gills. And they'll get essential nutrients from the fish, and they'll stay on there for a couple weeks or a month, depending on the temperatures and the mussel species. And once they have developed, they'll drop off as a juvenile mussel. And here's a picture of some juvenile mussels in a lab. They're still very small and they're not filter feeding yet. So after about a month, the organs will kind of transition inside, and then they'll begin filter feeding and will basically look like a smaller version of an adult mussel. So mussels can be categorized as either generalists or specialists, depending on the type of host fish that they use. In this picture, examples A, B, C, and D, these are examples of mussel specialists. So as you can see, they have these lures and they're trying to attract a fish. And a mussel specialist can only transform their glaucidia on a certain fish species or a fish species in a certain family. And so it's really trying to attract that fish. And if it attracts the wrong fish, it can actually be a waste of glaucidia. So the glaucidia will still attach to the fish, but then they won't transform into a juvenile. So in examples E and F, these are mussel generalists, and they broadcast their glaucidia. So in E, this mussel is just releasing its glaucidia up into the water, and this is in the hopes that a fish will swim through and then the glaucidia will attach. In the example of F, these are actually mucus strands which contain the glaucidia, and this is a type of passive entanglement where it's probably trying to attract a fish that's using a certain space in the water column, and when the fish swims through, the glaucidia will attach. So here in Massachusetts, we have about 12 species. This is a little bit different than some of the river systems down south that can have up to 100 species in the Tennessee River, for example. But even though we don't have as many species, they are really representative of a lot of different habitats, which is really interesting. So here in the Connecticut River watershed in the main stem, there's the yellow lamp mussel. There's also the dwarf wedge mussel, which is the one federally endangered species under the U.S. Endangered Species Act. The other, about half of these are protected under the state, so they're endangered or a species of concern. The eastern elliptio is a mussel that a lot of you might have seen before. It's the common species, and it occurs in a wide variety of different habitats. And if you've ever gone boating in the Connecticut River and stepped out of your boat onto the muddy bank and have just seen mussels everywhere, it's most likely these eastern elliptio. So there's also a common misconception that freshwater mussels are endangered because you see them so often, but we do have a lot of different species. And unfortunately, images like this are becoming more common, and they're actually making the news. And these are results of massive mussel die-offs. There's actually a task force assigned to try and help understand these. Here in the state, there has been local extirpations of rare mussels, and then also even within populations, we're seeing declines in the number of individuals within populations. So to kind of put it bluntly, we are really just living through a global mollusk crisis. This is happening in our lifetime. North America, the most speciose, we have 300 species, and yet 213 of them are listed as endangered, threatened, or locally are at risk. That's 72%, it's a really high number. And then just within the last 100 years, about 30 of these taxa have become extinct. And if anybody has any reasons of why this is, it's mainly due to the modifications that we make to the environment around us. So here's an example of a river that is experiencing a lot of trauma. There's no more riparian area here. There's some clear habitat loss, and this can be really important for providing vegetation, which can influence the shade in the river, can also be a great connection of nutrients between the river and the bank. But what habitat loss, and in this instance parking lots, are really good at is providing runoff to the river. So it's a great way to put pollutants into our rivers. So any kind of a dumpster here or a car that's leaking oil, all those things can enter our freshwater systems. Also going on here is some channelizations on both sides of the river. So this is preventing any overbank flows during high waters. Instead, it's kind of increasing the velocity and the bottom of the river, and this can influence the substrate and the invertebrates that you find there. It can also influence the fish communities. And then, of course, dams. So dams as we just learned, mussels are dependent on fish, and so dams can limit the movement of fish upstream and downstream, so it can really fragment mussel populations and genetic exchange between them. It can also limit dispersal into new habitat that could potentially support mussels. And this is just on top of all the other kind of side effects that dams provide to the environment, which these negative effects are changes in flow or temperature, and these can really alter even the fish communities and the invertebrate communities. So as humans, I think that we should all inherently care about biodiversity loss, but aside from that, freshwater mussels are also ecologically important. They filter our rivers up to 8 gallons a day for one mussel, and that depends on the size of the mussel and the species as well as the flow rates. But another unique thing is that when there's these really dense mussel beds, there's the ability to support microbes that can remove nitrogen from our rivers, and so excess nitrogen can enter our systems through a lot of different human activities, and there's not too many processes in the rivers that can directly remove it. They also increase the overall biodiversity by supporting other macroinvertebrates, which can be food for fish. And then they anchor into the substrate, so their foot in these dense mussel beds actually provides stability to the overall system. So here's a picture of one special mussel. It's called the Brook floater, and it's what my dissertation is focused on, and so I'll talk about this mussel for the rest of today. So here's a map from NatureServe, which shows the distribution of this mussel. It occurs from Georgia to Canada and New Brunswick and Nova Scotia. And the red just means it's critically imperiled, and then the yellow is imperiled, and the brown is not listed. But these records are very different than this map of Massachusetts. So here in Massachusetts, Barry Wicklow from St. Anselm College in New Hampshire, who has been studying brook floater for a really long time, he compiled a lot of historical records and worked with Mass Wildlife and the Natural Heritage and Endangered Species Program that keeps these records and updates them. And he found that brook floater in the state used to be in 10 watersheds, and now they're only in 4. So the red are places where they used to be found, and now they're only in the west branch of the Farmington River, Bachelor Brook, and the Ware River, which is within the Connecticut River watershed, and then the Nissitissit River in the Nashua River watershed. So my research has really been focused on trying to develop a way to restore this mussel. And I've kind of broken it down into 4 steps, and I will just briefly go through these. The first step was trying to understand where we find this mussel. So what is their habitat that they're in right now? And then with the help of volunteers in CRC, we were able to collect a lot of data for that. And then I'm working on creating models that will predict where we find them based on these environmental variables. The second step is propagation. So that's in a lab, going out and collecting mussels that have Glaucidia, and then collecting fish that we think are hosts and bring them into the lab and putting them together, and then growing mussels in that environment. And then once we have those mussels, figuring out how to keep them alive. There's a really high mortality when they're younger. And so trying to figure out their growth and survival can be challenging. And then growing them large enough where we can then put them back into our rivers. And so that's a whole other question of identifying habitat where we think could support these mussels again, whether that's augmenting or adding to current populations, or looking in other watersheds, maybe historical watersheds that used to have them, that could support the mussels again. And so one step that needs to be in first is to assess the water quality in these rivers. So for this first step, the habitat component, we would go out to a river, and with the help of volunteers, we would survey the stream and collect different measurements and also mussels to identify. And here's just an example of that. So this would be 6 observers, and we would work our way upstream collecting these measurements and snorkeling or using a clear bottom view bucket. And then some of these larger river systems, you need a lot of people. So it's really, the core volunteer group that we got was really essential in collecting this data. It was also probably one of the most, it was the best part of the project, basically. It was really fun. So I'm still working on these habitat variables, but these habitat models, but one variable that keeps showing up is that the substrate is important, or the size of the pebbles. So if you came out on any of these surveys with me, you remember measuring pebble widths, and it turns out that the size of the substrate is really important. And this isn't necessarily new, so a lot of other people who have modeled habitat for freshwater mussels have found that the substrate is one of the most important aspects to predict abundance. So here we have a graph, and this vertical axis is called the y-axis, and on it we have Brook floater, and CPUE is just the catch per unit effort. So we're constantly timing the areas that we search. So this number of Brook floater that we find is just standardized by the amount of time that we search. And on this axis, we have pebble size diversity. So this just means that where there's no diversity in pebble sizes, where it's 0, that could be all cobble or all sand. It's just all one size of sediment. And we don't find as many Brook floater in that type of sediment. As this increases in diversity, so sand and small rocks are incorporated, or small cobble, then we start to see more Brook floater, but there's still some variability in the substrate composition. But as this pebble size diversity increases and you have larger boulders and then sand like this, that's when we don't find as many mussels. So this is probably an artifact of the type of flow conditions that have created the substrate. The mussel or Brook floater still needs to be able to bury themselves into the substrate and also shelter during high flow conditions. So the sediment bed needs to be really stable to support them. And that's also true with a lot of other mussels too. So the second component, called propagation, which I mentioned earlier, is often recommended in recovery plans for endangered mussels. And the idea is to grow mussels so that we could then put them back out. And if these are added to populations as an augmentation, and this basically just increases the density of the number of mussels in a certain area, which can help to increase the probability of reproduction. Another approach is to reintroduce mussels into historical ranges, but this can be tricky because you don't want to put them in habitat where they will die. So you want to only consider these watersheds if you think that the habitat has improved enough to support them again. And all of this propagation work that we did was at the Cronin Aquatic Resource Center, which is a Fish and Wildlife Service center in Sunderland, Massachusetts. It used to be a salmon hatchery, and then it was converted into a mussel propagation facility. So the objective of my work was to try and figure out what host fish, Brook floater would be the best to grow them on a large scale so that we could then put mussels back out into streams. And fortunately, there was already some great host fish work done, and this was by Barry Wicklow again. And he found that the mussel was a generalist, so it could transform on a lot of different species of fish in different families. And there was more host fish work done by Chris Eads in North Carolina, who found that bluegill and Piedmont darter were a really great host. But probably the most interesting thing is even though both studies found that brook floater was a host generalist, when they tested the same species, in some instances it would be a host in one location but not the host in another location. So we really weren't sure what fish to use when starting for propagation in Massachusetts, but we did select some of the fish that they had already been tested. This is a picture of a whitesucker, and one additional result that Barry found was that on top of the variability in between fish species, that the younger whitesucker was able to transform Glaucidia into juveniles, but then the older whitesucker wasn't, so there's also an age difference. So we basically started this project for propagation by going out and collecting these adult female mussels that were holding Glaucidia, and this is a picture of 110, and bringing them into the lab. Brook floater have these orange colored foot, and then we would induce the mussel to release its Glaucidia. So these are all the little white dots, and they are in mucus strands, so that's their mode of reproduction. And then we would take this Glaucidia and we would add it to a bucket with fish, and we would put an air stone in there and hope that the Glaucidia attached to the fish. So here's a picture of the Glaucidia attaching to the fish gills. And if we look, this is a kind of zoomed in photo of a Glaucidia, and they have this hook right here. And this hook can embed in the fish, and so they can actually attach all over the fish as well. But what we were really aiming for was to find a host that would support a lot of Glaucidia attaching to it, but then a lot of those Glaucidia that are on the fish to then transform, because we know it can be different between species. So we would then put these fish in tanks and then collect the number of juveniles that dropped off of the fish. So here's just a picture of the juveniles with their with this clear foot right there. They're still very small. So for this project, we tested 10 different fish species: slimy sculpin, long-nosed dace, black-nosed dace, brook trout, brown trout, rainbow trout, Atlantic salmon, bluegill, whitesucker, and banded killifish. And we found that slimy sculpin was probably the best fish to move forward with large-scale production, but we also found a lot of differences between experiments, especially with the brook trout. In one instance, it would transform a lot of Glaucidia into juveniles, and then in another experiment, it didn't perform as well. And so we're not sure if that has to do with where we collected the fish from, or where we collected the mussel broodstock from, or even an artifact of lab conditions. But it's clear that we still have a lot to learn about the host fish that mussel generalists use, like the brook floater. And then even though, you know, these fish were able to transform Glaucidia in a lab, doesn't necessarily mean that the mussel is using these fish in the wild as hosts, because in the lab we're really uncoupling those ecological barriers that they might experience otherwise. So that just includes the actual ranges of and the habitat overlapping of the fish and the mussel, and then also the fish being exposed directly to the Glaucidia So where the fish is in the water column and how it's feeding. That might be conducive in the wild to it not being a host. So from here we would try and grow these mussels in a lab. We tried different feeding, different types of food, different types of substrate. And we ended up with two cohorts of mussels. One was 2 years old and one was 8 months old. And we took these mussels and wanted to put them back into the river, but we first did kind of an ultimate test of water quality. So these are called mussel silos. They're concrete domes and they have a chamber in the center, and this supports water flowing through the chamber. Here's just the little mussels in the bottom. And we decided to put these out into the 4 rivers where we still have brook floater populations to compare the growth and survival between these rivers, and also the really small cohort. It'd be interesting to see if these lived, because when we do these types of mussel surveys, we can't find mussels that are that small. The detection is too low. So in each of these rivers, we would put 4 mussel silos, and this is just a look in the chamber with the mussels, 2 different size classes. And we'd visit these mussel silos every 2 weeks and take measurements, and also take a lot of different water quality variables. And we found that there was a lot of growth in the river that was actually kind of has the poorest population. So at week 0, this mussel was 9.5 millimeters, and then by week 16, it was 19.1 millimeters. And this was a really high growth rate, and so we're not sure if this is actually normal or just an artifact of excess nutrients that are entering this river and creating more food availability. But the good news was that we did find really high survival of these mussels in all 4 of the rivers. And we also know that the mussel silo itself can influence differences in growth, because the water flow through the silo influences how much food is being delivered to the mussels. So I didn't, during my dissertation, there's still a lot of things left unanswered about mussel restoration that still need to be addressed when considering releasing mussels back out into rivers. And these include just genetic differences between populations. So from where you're collecting the brood stock from and where you're planning on putting the mussels out, if you would like genetic mixing, or if you want the populations to stay separate. And also bacterial or viral infections, parasites between that can be exchanged between mussels and fish when releasing lab propagated mussels into natural occurring mussel beds. Stream connectivity we know is important for establishing new populations, so that's considering the number of dams and culverts in a river where you're going to put them, and then the fish presence or density of host fish and the species that are there. And then lastly, the resiliency of that river reach to climate change. So this is just how it will be affected by long-term warming and extremes in flow, whether that's drought periods and dewatering or high floods. So a lot of people participated in this project, and a huge thank you to all of them, including all of the great volunteers that came out. And if you have any questions that you don't want to ask today, you can feel free to email me. My email is down here. All of this work was funded by 2 grants. The first was the Massachusetts Environmental Trust and their sale of license plates. So if you see the plates with the right whale tail or the leaping brook trout and the Blackstone Valley Mill, and I think there's a new one with a striped bass too. If you have one of those license plates, thank you for contributing to the project. and also a larger grant, the U.S. Fish and Wildlife Service State Wildlife Grant. So with that, I think I'm out of time and happy to take questions.
Wonderful. Wow, Ayla, that was so fascinating. I learned a ton. And while people are starting to type in their questions, I just wanted to ask one back to the beginning when you were showing how the mussels are releasing the eggs and whether they attach to fish or not. The ones that just float them out there, is there any data on what percentage of them actually find a host? Or can you talk about that process of whether they attach or not, or what happens when they don't? I'm assuming they just die off, or do they land somewhere else and live somehow?
That's a great question. They're really hard to study naturally in the wild, and especially to witness the events of mass release like that is pretty special. So I don't know if there's any data on how many of those actually attach to fish versus fall to just the sediment bottom and eventually become unviable. But they can survive for a little bit of time outside of the mussel, and it kind of varies depending on the species. But if it doesn't attach right away and it's on the sediment bottom, then it probably wouldn't encounter a fish from there either. But that strategy is pretty unique too, because it's also what some marine bivalves use. So if you ever see pictures of those like mass release events of their larvae, so it's kind of cool.
Great, thanks. Okay, so I'll start going through some questions now, and folks, please continue to throw them in the Q&A box. So here's one from someone who lives in Vermont and has been coming to the same lake for his whole life. And as a boy, never saw a single mussel. And in the last 20 to 30 years, they are now appearing. So he has multiple questions in this question about how did they get there? How did someone bring them in? Could they have been brought in on the gills of fish? Can you speak to those, how mussels would suddenly appear in a new place?
Cool. I'm curious what kind of mussel you're referencing, if it's a native freshwater mussel. I guess it kind of depends on the mussel. Hopefully it's not a zebra mussel. But yeah, they can be transported on fish, and even fishermen who bring in other fish for bait, they can be on the gills. And if the water quality and the food availability has changed in the lake where it can support the freshwater mussels there, that could be what's going on. But yeah, it'd be interesting to know what kind of mussels they're referencing.
Okay, that may be hard to find out, but if that person can do a little more digging.
Or if they want to send me pictures too, like they can email me pictures, that's fine.
Great, okay. The next question is, what are the top native predators for freshwater mussels? Otters, carp, catfish?
Yeah, so definitely listed some there. Otters feed on them. They can actually find mussel middens along the rivers where there's just a bunch of shells, and those are from predators feeding on them. Raccoons can also be predators, sometimes crayfish too, and then definitely some other fish species will eat mussels, especially after they have, especially after they've died too. They'll feed on the tissue inside the shells. But I think it's interesting because predators I don't think are as studied now because of the massive declines in freshwater mussels. It's a lot of the research now is focused on how human-related impacts are influencing the declines in mussels, and less so on the native predator aspect.
What water quality parameters have you tracked? Which seem most important to survival, and what is the tolerable temperature range?
It's a great question. So especially with the silo study, we were looking at nutrients like total phosphorus and total nitrogen. And then also, for example, calcium. We know that they need calcium to build their shells, and alkalinity, which is the buffering capacity of the river. And so those are all really important parameters to look at. And we also were looking at chlorophyll A as a representative of the amount of food that's available in the river. And we didn't look at any other basically pollutants, which is something else that's interesting, and especially it would show up in a lot of rivers in eastern Mass too. And the tolerable temperature range varies based on species, and it's something that I think we still have a lot to learn about. So we put these things called temperature loggers out into rivers, and they record the temperature every 15 minutes. And then we can graph this and see how the temperature is changing over time. And in the future, in some of these rivers that are experiencing really low water events and high warming, this might be something that's impacting their survival. But as far as the temperature thresholds that are causing mortality, we don't know that for a lot of species, what that specific threshold is.
Okay, thanks. Back to predators. Do mink or fisher eat mussels, and how about loons?
Yes, I think mink or fisher can eat mussels. I don't know about loons. I would imagine not, but that's purely a guess. So I could look that up, see if anybody's ever looked into that.
Great. Well, that seems to be all the direct questions we've been asked. Ayla, do you have any other thoughts you want to share? And otherwise, I can close it out.
Yeah, thanks for giving me this platform to talk about freshwater mussels. It's something that we just don't hear about all the time. So again, feel free to email me any other questions that come up, and I can also include some links to some videos of fish attacking mussel lures, which are fun.
Great. The images you shared were fantastic. I love all those photos. I wonder how people get them. So just want to take this moment to really thank Ayla for her time and all this incredible research and writing that she's doing about this critical brook floater mussel. And thanks to all of you who came today to share your excitement and enthusiasm for river health. So just so you know, this has been recorded, so we'll be posting it on our website at ctriver.org/livestream within the next 24 hours. You can watch it again or tell your friends. And you'll get a follow-up email with Ayla's contact info and all of the links that I've been talking about, and a list of the following live stream episodes. So we hope you'll join us again. And basically, we're really happy to be able to share our work with you in this way. So come again and keep your energy high, and we hope to see you again soon. Thank you, everybody. Have a good afternoon.
Thanks, Stacey.